हेलो एवरीवन, वेलकम बैक टू माय चैनल। आज हम पढ़ेंगे चैप्टर 9 ऑफ क्लास 11th बायोलॉजी, बायोमोलिक्यूल्स। इसमें पढ़ेंगे हम हाउ टू एनालाइज केमिकल कंपोजिशन, प्राइमरी सेकेंडरी मेटाबोलाइट्स, बायोमैक्रोमोलिक्यूल्स, प्रोटीन्स, पॉलीसेकेराइड्स, न्यूक्लिक एसिड, स्ट्रक्चर ऑफ प्रोटीन्स एंड एंजाइम्स। सो लेट्स स्टार्ट दिस. There is a wide variety in living organisms in our biosphere. Now, a question that arises in our mind is, are all living organisms made of the same chemicals, elements and compounds? You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissue or a microbial paste, we obtain a list of chemicals like carbon, hydrogen, oxygen, and several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of earth crust as an example of non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of earth's crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in earth crust. How to analyze chemical composition? We can continue asking in the same way what type of organic compounds are found in living organism. How long does one go about finding the answers? To get an answer, one has to perform a chemical analysis. We can take any living tissue, a vegetable or a piece of liver, etc., and grind it in trichloroacetic acid, Cl3CCOOH, using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called filtrate, or more technically the acid-soluble pool, and the second, the retentate or the acid-insoluble fraction. Scientists have found thousands of organic compounds in the acid-soluble pool. ये कुछ एलिमेंट्स के परसेंट वेट ऑफ अर्थ क्रस्ट बाय ह्यूमन बॉडी दे रखा है, आप देख सकते हैं. जैसे कार्बन का 0.03 होता है 18.5. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extract the compound, then subjects the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolate and purifies a compound. Analytical techniques, when applied to the compound, give us an idea of the molecular formula and the portable structure of the compound. All the carbon compounds that we get from living tissues can be called micromolecules. Biomolecules. However, living organisms have also got inorganic elements and compound in them. How do we know this? A slightly different but destructive experiment has to be done. One weigh a small amount of a living tissue, say a leaf or liver, and that is called wet weight, and dry it. All the water evaporates. The remaining material gives dry weight. Now the tissue is fully burned. All the carbon compounds are oxidized to gaseous form, CO2, water vapor, and are removed. What is remaining is called ash. This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid-soluble fraction. Therefore, elemental analysis gives elemental composition of living tissue in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives an idea of the kind of organic and inorganic constituent present in living tissues. From a chemistry point of view, one can identify a functional group like ketones, aldehydes, aromatic compound, etc. But from a biological point of view, we shall classify them into amino acid, nucleotide bases, fatty acid, etc. Amino acid are organic compounds containing an amino group and an amino group as substitute on the same carbon, that is the alpha carbon. Hence they are called the alpha amino acids. They are substituted methanes. There are 4 substituent groups occupying the 4 valency position. These are hydrogen carboxyl group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R group in this proteinaceous amino acid could be hydrogen. The amino acid is called glycine. A methyl group, alanine, hydroxyl methyl, serine, etc. 3 of the 20 are shown in figure 9.1. The chemical and physical properties of amino acid are essentially of the amino, carboxyl, and the R group functional groups. Based on number of amino and carboxyl group, there are the acidic, glutamic acid, basic, lysine, and neutral, valine amino acid. Similarly, there are aromatic amino acid, triosine, phenylalanine, tryptophan, phenylalanine. A particular property of amino acid is the ionizable nature of NH2 and COH groups. Hence, in solutions of different pH, the structure of amino acid changes. ये आप देख सकते हैं NH3 है, कार्बन है, एसिटिक एसिड है, ये R ग्रुप है, A यहाँ पे चेंज हुआ, फिर यहाँ पे चेंज हुआ, B इज द ज़्वेटरानिक फॉर्म है इसका. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be methyl, CH3 ethyl, C2H5, or a higher number of CH2 groups, 1 carbon to 19 carbon. For example, palmitic acid has 16 carbons, including carboxyl carbon. Arachidonic acid has 20 carbon atoms, including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more CC double bond. So, saturated किन्हें कहते हैं हम फैटी एसिड्स को जिसमें डबल बॉन्ड नहीं होते हैं, यानी सिंगल बॉन्ड वालों को हम सैचुरेटेड कहते हैं. तो आप इसे ऐसे याद रख सकते हैं कि इसमें स्पेलिंग अनसैचुरेटेड सैचुरेटेड में किसकी छोटी है, सैचुरेटेड ये तो यानी सिंगल बॉन्ड होगा. अनसैचुरेटेड में डबल या ट्रिपल बॉन्ड होते हैं. अनदर सिंपल लिपिड्स इज ग्लिसरॉल व्हिच इज ट्राईहाइड्रोक्सीप्रोपेन. Many lipids have both glycerol and fatty acid. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. They are also called fats and oil based on melting point. Oils have lower melting point, example gingelly oil, and hence name as oil in winters. Can you identify fat from the market? Some lipids have phosphorus and a phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases, adenine, guanine, cytosol, uracil, and thymine. We found attached to a sugar, they are called, when found attached to a sugar, they are called nucleosides. अगर शुगर अटैच्ड हो तो उन्हें न्यूक्लियोसाइड्स कहा जाता है. इसको हम हाईलाइट कर लेते हैं. इसे भी हम हाईलाइट कर देते हैं. Adenosine, guanosine, thymidine, uridine, and cytidine and nucleosides. Okay, तो sugars हैं basically ये adenosine, guanosine, thymidine, uridine, and cytidine. जब आप शुगर के साथ फॉस्फेट ग्रुप होता है तो ये वो है एडिनलाइक एडिनलिक एसिड, थायमिडिक एसिड, ग्लूकोलिक एसिड, यूरिडाइलिक एसिड एंड साइटालिक एसिड एंड न्यूक्लियोटाइड्स. Nucleic acid like DNA and RNA consists of nucleotides only. DNA and RNA function as genetic material. ये भी important है. तो न्यूक्लियोटाइड्स किसे कहते हैं? जैसे आरएनए डीएनए उनमें सिर्फ न्यूक्लियोटाइड्स होते हैं. एडेनाइलिक एसिड हो गया, थायमिडिक एसिड हो गया, ठीक है. साइड्स अलग होता है और न्यूक्लियोटाइड्स अलग होता है. तो आरएनए डीएनए में क्या होता है? टाइड्स. आप ऐसे याद रख लेना. टाइडी वर्क होता है आरएनए डीएनए का, ठीक है. ये अब डायग्राम देख सकते हैं. शुगर कार्बोहाइड्रेट, ग्लूकोज हो गया, राइबोज हो गया, C6H12O6, C5H10, तो एक कार्बन कम होता है. अमीनो एसिड हो गया, ग्लाइसिन, ये पेज इंपॉर्टेंट है. एलेनिन, सिराइन, ग्लाइसिन, एक कार्बन, नाइट्रोजन, एसिटिक एसिड. एलेनाइन, एक कार्बन, दो कार्बन, नाइट्रोजन, एसिटिक एसिड. सिराइन, 3 कार्बन, 2 कार्बन, अल्कोहल और ओइक एसिड. फैटी एसिड, पाल्मिटिक एसिड हो गया, ग्लिसरॉल हो गया. ग्लिसरॉल का फॉर्मूला 3 कार्बन एंड ओइक, CH3 वो पाल्मिटिक एसिड. ट्राइग्लिसराइड हो गया, R1, R2, R3 फैटी एसिड्स. फॉस्फोलिपिड लेसिथिन हो गया, फैट एंड ऑयल्स हो गया. एडेनाइन हो गया, प्यूरीन. एडेनोसाइन हो गया, एडेनाइन OH. एडेनलाइड, एडेनसैलिक एसिड हो गया. न्यूक्लियोसाइड्स, नाइट्रोजन बेस हो गया, NH हो गया, OO, यूरेसिल पैरामिडीन. न्यूक्लियोसाइड्स, यूरेसिल OH. न्यूक्लियोटाइड्स, ओके. सो प्राइमरी सेकेंडरी मेटाबोलाइट्स क्या होते हैं? The most exciting aspect of chemistry deals with isolating thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If we were to make a list of biomolecules, such as list would have thousands of organic compounds having amino acids, sugars, etc. For reason they are given in section 9.10. We call these biomolecules are metabolites. Pigments. In animal tissue, one notices the presence of all such categories of compound. जैसे 9.1 में दे रखा है. These are primary metabolites. However, when one analyzes plant, fungal and microbial cell, one could see thousands of compounds other than these primary metabolites. Example: alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigments, sand, gum, spices. These are called secondary metabolites. तो यहाँ से लेके यहाँ तक important है. When primary metabolites have identifiable function and play known roles in animal physiological process, we do not at the moment understand the role or function of all the second metabolites in host organisms. However, many of them useful to human welfare, जैसे rubber, drugs, spices, sands, pigment. Some secondary metabolites have ecological importance. In the later chapter years you will learn about it. Some secondary metabolites जैसे pigment हो गया, alkaloids हो गया, terpenoids, essential oils, toxin, lens, lectins, drugs, polymeric substances. Pigment carotenoids, anthocyanins, alkaloids, morphine, codenine, terpenoids, monoterpenes, diterpenes, essential oil, lemongrass oil, etc. Toxin, aberrant, ricin, lens, lectins, okay, drugs, polymeric substances हो गया. Biomacromolecules. There is one feature common to all those compounds found in the acid-soluble pool. They have molecular weight ranging from 18 to 800 daltons approx. The acid-insoluble fraction has only four types of organic compounds, that is proteins, nucleic acid, polysaccharides, and lipids. These classes of compound, with the exception of lipids, have molecular weight in the range of 10,000 daltons and above. For this very reason, biomolecules, that is the chemical compound found in living organism, are of two types. One, those which have molecular weight less than 1000 dalton and are usually referred to as micromolecules or simply biomolecules, while those that are found in the acid insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids whose molecular weight do not exceed 800 dalton come under acid insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell, membrane, and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid-soluble pool represent roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid-insoluble fraction. Together, they represent the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we observe that the water is the most abundant chemical in living organism. Proteins. Proteins are polypeptides. They are linear chains of amino acid linked by peptide bonds. Each protein is a polymer of amino acid. As there are 20 types of amino acid: alanine, cytosine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is important as later in your nutrition lesson. You will learn that certain amino acid is essential for our health and supplied through our diet. Hence, dietary proteins are the sources of essential amino acid. Therefore, amino acid can be essential, non-essential. The later are those which our body can make. While we get essential amino acid through our okay, so जो नॉन एसेंशियल होते हैं वो हमारी बॉडी लेटेड है नॉन एसेंशियल मेक ज़रूरी नहीं है खुद बना लेती है मेहनत करने की ज़रूरत नहीं है बाकी एसेंशियल जो होते हैं जो डाइट के लेने पड़ते हैं some transport nutrient across cell membrane, some fight infectious organism, some are hormones, some are enzymes, etc. Collagen is the most abundant protein in animal world and ribulose, biphosphate carboxylase, oxygenase. Rubisco is the most abundant protein in the whole of the biosphere. तो कौन सा है abundant? Rubisco. Okay. Next is polysaccharide. The acid insoluble palate also has polysaccharides. Carbohydrate is another class of macromolecules. Polysaccharides are a long chain of sugars. They are thread, literally cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Polysaccharides. The acid-insoluble also has polysaccharides. Carbohydrate as another class of macromolecules. Polysaccharides are long chain of sugar. They are threads, literally a cotton thread containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharides consisting of only one type of monosaccharide, that is glucose. Glucose is a homopolymer. Starch is a variant of this, but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Insulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right hand is called the reducing end and the left hand is called the non-reducing end. It has branches as shown in the figure in the form of cartoon. Starch form helical secondary structures. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helicals and hence cannot hold I2. As you can see, ग्लाइकोजन का एक रिप्रेजेंटेशन दे रखा है. Plant cell walls are made up of cellulose. Paper made from plant pulp and cotton fibers is cellulosic. They are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars. Example, glucosamine, N-acetyl, galactosamine, etc. Exoskeleton of arthropod, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. Nucleic acid. The other type of macromolecule that would find in the acid-insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. These comprise the true macromolecular fraction of any living tissue or cell. For nucleic acid, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third is a phosphoric acid or phosphate. As you can notice in figure 9.1, the heterocyclic compound nucleic acid are the nitrogen bases named adenine, guanine, uracil, cytosine, thymine. Adenine and guanine are substituted purines, but the rest are substituted pyrimidines. This is important. Skeletal heterocyclic ring is called purine and pyrimidine respectively. The sugar found in polynucleotide is either ribose, a monosaccharide pentose, or 2-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid DNA, while that which contains ribose is called ribonucleic acid RNA. So we continue in the next video. Please do like and share and subscribe.